Hello guys, I am Diktesh, currently studying BMLT in MS Company. So today in this video, we will be discussing about a very important topic from the point of view microbiology practical exam and as well as also from the theory. That is gram staining. Don't worry, this is going to be a short video. So let's start gram staining. So what is gram staining? Gram staining is a basic routinely used microbiological staining to differentiate between the gram-positive organisms and gram-negative organisms. Modification is a staining technique that was originally developed by Hans Christian Gram. Gram stain is named by this scientist. Let's discuss about the procedure. What is the procedure of the gram staining? First, we have to fix our slide. So when we are making the slides from the sample, we have to fix it by heat fixing. The smear made on the slide from bacterial culture or specimen is air dried and then heat fixed. Then comes the step 1, that is primary staining. We are using crystal violet as a primary stain. Over the slide we putting crystal violet. So the smear is stained with dyes such as crystal violet. If crystal violet is not available, then we can use gentian violet or methyl violet, and the time is 1 minute. So first step is crystal violet for 1 minute. Then we have to wash it with water, not like that you hold the slide and directly the water comes on the smear. You have to slowly rinse the slide so that the slide not washed out with the water flow. So crystal violet stains all the bacteria violet in color, irrespective of the gram positive or gram negative. Then step 2 is mordant. We are using mordant grams iodine. Grams iodine is poured over the slide for 1 minute. Then after 1 minute again the slide is rinsed with water. Grams iodine act as a mordant, binds to the dye to form bigger di-iodine complexes in the cytoplasm. So next step, third step is decolorization. In the next step is pouring a few drops of decolorizer to the smear, and slide is immediately rinsed with water. Decolorizer removes the primary stain from the gram-negative bacteria, while the gram-positive bacteria retains the primary stain. Why this happens, I will explain when I will be explaining the principles of gram stain. For now, you just understand that the decolorizer, after decolorizer, gram-negative bacteria will decolorize, and gram-positive bacteria that will retain the primary stain color. It is very important that decolorizing should not be over decolorized. It is very specific and you have to maintain the time very specifically. We can use decolorizing agent as acetone or ethyl alcohol or acetone alcohol, and the timing is different for different chemicals. If we are using acetone, then it should be 1 to 2 seconds only. If we are using ethyl alcohol, then 20 to 30 seconds, and if we are using acetone alcohol, then it can be around 10 seconds. So suppose this is a slide, and if you are putting decolorizing over it, so you have to just put the decolorizer and wash it. Not like that you are putting the decolorizer waiting for the time you don't know. Not like that. This is the slide. You put the decolorizer and immediately after 1 to 2 seconds. That means you are putting that and washing it like that. And step 4 is counter staining. Counter staining, secondary stains such as safranin or dilute carbolfuran is used. It is added for 30 seconds, and after 30 seconds again it is washed and dried in the air and seen after oil immersion objectives. So what we will see after the staining is over, how the gram positive bacteria will look like and the gram negative bacteria will look like. So after gram staining, gram positive bacteria will look in color violet, okay, and gram negative bacteria appear pink. So this is our gram negative bacteria, and this is also gram negative. This is gram positive, and this is positive. This is basically gram positive bacilli, and this is your gram negative bacilli. This is gram negative cocci. This is gram positive cocci. Okay, next come to the principle. Now, for the principle, you have to know the structure of gram positive bacteria and gram negative bacteria. So this is the structure of gram negative bacteria and this is gram positive bacteria. What is the main difference between gram positive and gram negative bacteria is the layer of peptidoglycan. So in the gram positive bacteria, peptidoglycan layer is very thick, and in the gram negative bacteria, the peptidoglycan layer is very thin. So here comes the theory, that is cell wall theory. So when we are adding, in the first step what we are adding? Crystal violet. So when we are adding crystal violet, that crystal violet penetrates both the cell wall for the gram positive and for the gram negative also. It is going in the periplasmic space, okay? The primary stain, crystal violet. Then that's why after the primary stain, both the bacteria is looking violet. Step number 2, we are adding mordant, iodine. What iodine is doing? Iodine is penetrating the gram through the positive bacteria, and here in the periplasmic space, that iodine and the dye is making a diiodine complex. Here also, gram negative also. Then we are adding decolorizing. So decolorizing agent is for the gram positive bacteria. Why it is not decolorizing? Because of the thickness of the gram, that is peptidoglycan layer. It is that peptidoglycan layer is very thick. So when we are adding decolorizer, it is not getting out from the cell wall. But in case of gram negative, as you can see, the peptidoglycan layer is very thin. So when we are adding decolorizer, that diiodine complex is washed out through the peptidoglycan layer, and it is very thin. So after that, in the last step, when we are adding safranin, that safranin is going through the cell wall in case of gram negative only, and stains that bacteria into pink. So after the staining, our gram positive bacteria is look like violet and gram negative is pink. So that is the basic cell wall theory, widely accepted theory. So let's come in the what of the theory is there. This is the cell wall theory that I have explained. Except that there is a pH theory, that is cytoplasm of gram positive bacteria is more acidic, hence it retain the basic dye for the longer time, and iodine serves as mordant. It binds with the primary stain to form diiodine complex, so it gets retained in the cell wall. So this is all about the gram staining. So from here, what questions can be asked? You have to know that, because it is very important topic in the practical aspect. So in the practical, you have been asked that the procedure of the gram staining. What is the procedure of the gram staining? After the gram staining, how the gram positive will look like and how the gram negative will look like. That is the second. Third thing is, what is the principles of gram staining? Then you have to tell about the pH theory, that cell wall theory. And there is a third theory, that is magnesium ribonucleate theory also. magnesium ribonucleate is present in the cell wall of gram positive. That's why it retain the primary state. That is also theory that you can read. And one more thing you have to read that is asked in the Viva, that is what are the modification of the gram stones. I can find that there is Koploff and Biermann's modification, Jensen's modification, Brown and Brenn's modification. This much, if you can tell, that is enough. And also, you have to remember that in the Koploff modification, what changes are there. So, the primary stem is using here, we are using methyl violet, and as counter stem, we are using basic fusion. In Jensen's modification is useful in case of meningococcal and gonococcal. There is the use of absolute alcohol as decolorizer, and in the Browns and Brenn's modification, it is used in the case of actinomycetes. So these are the things about the gram staining. Thanks for watching.